We have a special challenge at Harvard that we've got an extraordinary number of outstanding investigators that are spread across many different sites that include the Mass General, the Dana-Farber, Beth Israel Hospital, the Harvard University Faculty of Arts and Sciences, the Harvard Medical School.
In order to really bring this all together, we need a juxtaposition of the genetics, the molecular biology, the developmental biology, and the bioengineering. It's in this kind of environment, the Harvard Stem Cell Institute, that expertise can be brought to bear.
It's a fairly altruistic institution. It was started by an investigator who was willing to give up money out of his pocket to start something good. And I think those of us who remember that feel like you should give something back.
What the HSCI does is to provide the most important incentive that you can provide, which is an opportunity to get together and meet with your colleagues to talk about problems of mutual interest.
It's promoted collaboration that never would have taken place otherwise.
The nature of science has changed, and the platforms have evolved, and the technology's gotten more sophisticated. It's become increasingly important to work together as a group. And it's a model that the high-energy physicists have had for a long time, that's driven in part by the need for access to technology.
You start out with technology, fundamental world-class technology in stem cell biology, world-class engineering, and our capabilities in heart stem cell biology, cardiology, and in what is known as translational research, where we take these basic science findings to the patient. This embodies the interdisciplinary spirit of the Harvard Stem Cell Institute.
There's been a very positive response to simply coming together in the same room on a monthly basis between literally dozens of laboratories around the city to talk about common biology. And what we find is the nervous system is so complex that people working in the eye didn't even know that some of the same molecules and pathways or approaches that they were pursuing were applicable in the brain or the spinal cord. And that has shortcut many long paths that people might have taken and brought light and gotten to discoveries much quicker by crosstalk between laboratories, between groups, between newfound friends, and then that can leverage across HSCI, where nervous system groups work with blood disease groups or diabetes groups and find that much of the biology is common and gets us to all these different goals more rapidly.
One collaboration was between myself and Dr. Humphreys, who's a faculty member here in Reno, and Dr. McMahon and Dr. Kobayashi and Valerius. That group has developed these mice that we can engineer in such a way that you could label up, that is, you can color in the mouse every cell of the functional unit of the kidney, either blue or red. And so that's actually helped us to define that the repairing cells were not coming from the interstitial region, but they're coming from the tubule itself. And that could only be done with the juxtaposition of our expertise and Dr. McMahon's laboratory. And that was brought together again by the HSCI.
We're doing a screen for genes involved in self-renewal using a technique called small interfering RNA or siRNA technology. And that technology was really developed in David Scadden's group, and we've never done it before, but one of my postdocs now is working with David's postdoc to learn the technique and then apply it to our system. And I don't think we would be doing that if it weren't for the blood program.
One example of internal collaboration is a 5-way collaborative project in the ALS and spinal cord injury direction, in which, for example, Paula Arlata, a new assistant professor who's studying the development of the corticospinal motor neurons that connect the brain to the spinal cord, has teamed up with Susan Demecki, who has traditionally worked in the brainstem breathing centers. But in those centers, she developed some molecular tools that allow Paula to find out exactly which stem cells give rise during development to individual types of neurons. And that's work that without this multidisciplinary, multi-investigator, multi-laboratory approach would not come to fruition.
We've always admired the work of Doug Melton over at the college, but to be able to actually have a project that allows us to work closely with him has been enormously exciting to me.
The Harvard area is incredibly rich in outstanding investigators in hematopoiesis and leukemia, which is the obvious direct correlator in terms of disease states. One thing that HSCI has done is further unite these different investigators in terms of working together.
In addition to Doug Melton, it's opened up ways for me to work with people at the Brigham and Women's Hospital, Dick Moss and his team. It has opened up collaborations here at Joslin that weren't taking place before. It's opened up collaborations at MIT with biomaterials people, collaborations at Massachusetts General Hospital. So the whole Harvard community is really being pulled into this project.
We are in a position in the kidney program to really benefit from not only some of the capabilities that the Stem Cell Institute has introduced to the environment, but also the intellectual capital.
Just that ability to have a focused, organized program that brings us all together with common interests has been, it's been revolutionary.